Hello, everyone, and welcome to today's Cryo-EM Current Practices webinar. You're joining the 3 NIH-funded National Cryo-Electron Microscopy Centers. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. I'm joined today by staff members from two of the three centers, including my colleagues here at NCCAT, Ed Eng and Kasahun Naselu. Uh, Joining from the Pacific Northwest Cryo-EM Center, PNCC, is Maurizia Mileto. We don't have a representative from the Stanford Slack Cryo-EM Center S2C2. They're very busy with a workshop today, but we'll give you a quick update about what's happening there. Before we give the floor to today's speaker, we'll get a quick update from each center on the resources we make available to the research community at no cost. For our main event today, NCCAT has invited today's speaker, Evan Wood, who's joining us from San Diego State University. And Kasahun is going to introduce him properly after our center updates. If you're joining us for the first time today, welcome. This webinar is a regular event we host usually the last Thursday of each month, and we highlight users of the National Cryo-EM Centers who generously share the tips and tricks that have enabled them to make use of cryo-EM in their research. Next month is an exception to our regular schedule because of the busy holiday seasons at the end of November and December. We'll have one final talk this year on December 4th. Professor Robert McKenna from the University of Florida, Gainesville will be giving a talk. We don't have the final title yet, but rumor has it the topic will be viral capsid structure and function. So that should be a nice way to wrap up the year. The registration link for that talk is already available on the events page at cryoemcenters.org, where you can also find recordings of all of our past talks. For final logistics today, if you do have questions about accessing the National Center resources, please feel free to use the chat to reach out directly to the panelists here from the centers. But for questions for the speaker during the talk, please use that Q&A box, and we will be moderating Q&A at the end of the talk. So please send those at any time using the Q&A. All right, so let's get started with today's program. So a quick set of updates from the center. So I am not from S2C2, but I'll give you a quick hello from S2C2. Like the other centers, they offer access to high resolution data collection instrumentation, as well as a robust screening and optimization program. They have applications for user training, and they also run a number of workshops. In fact, right now they're running a workshop on data on model building and validation. And you can find all the information about what's going on at S2C2 on their website. And next, I will turn over to Maurizia to let us know what's new at PNCC.
Thank you, Christina, and good morning, everyone. My name is Marzia Miletto, and I'm a microscopist at the Pacific Northwest Center for Cryo-EM in Portland, Oregon. And I would like to briefly talk about what we offer. You can access PNCC through two different types of proposals. One is a screening access, which is aimed at low-resource institutions. And this proposal allows you to come on site for up to 5 days of sample prep and screening with staff. And the second type is a general access, which awards you up to 480 hours of microscope time a year for 2 years. And this can be used for both screening and data collection. So we do, so for general access proposals, we also offer an extensive training program. We offer a variety of workshops that cover the entire cryo-EM pipeline. And both type of proposals are assigned a PNCC staff microscopist as scientific point of contact, or SPOC. And this person can help you with questions about sample prep, data collection strategies, and so on. So we do have a lot of instruments, 6 microscopes, an Enterica, a Glacius 2 and 4 Krios. We both have Falcon 4i detectors, biocontinuum HD, and KT detectors, both equipped with either Select 3X or biocontinuum HD energy filters. And we do have a lot of instruments as well for sample prep. So VitroBots Leica GP2, VitroJet manual plungers. And so if you're interested in working with us, please go to pncc.labworks.org and apply. So you know our deadline for proposals review is the first of every month. And you will find all the information on the website. But please reach out with any questions. We will be happy to help, and we look forward to hearing from you.
Thank you, Maurizia. Next, I'll turn it over to Ed to let us know what's new at NCCAT.
Hi, I'm Ed, coming from NCCAT with my colleagues here in New York City. We offer high-end data collection on one of our 5 Krios instruments, grid preparation and screening, as well as cross-training. On the training front, this has been a good year for short courses. Earlier in the year, we kicked off with a tomography short course. Next week, we're actually having a micro-ED short course, and applications are open for our March 16th to March 20th, 2026 single particle short course. Talking about 2026, now is the time for society awards. If you're part of the Microscopy Society of America, we're accepting nomination for MSA Fellows class of 2026, as well as other society awards. That deadline is actually tomorrow, October 31st. If you're a part of the Biophysical Society, the cryo subgroup is welcoming nominations for the June Almeida Award. June Almeida was a virologist who first identified the human coronavirus and so far named after the corona parents of the virus particles. And in her honor, the cryo subgroup is offering that award. Nominations are due November 14th, and the awardee will speak at the cryo subgroup Saturday, February 21st in 2026. BPS 2026 will be in San Francisco. So if you'd like to learn more, you can go to nccat.nysbc.org. If you want to learn more about the societies, please let me know in chat.
Thank you, Ed. I will go ahead and stop sharing and ask Kasahoon to introduce our speaker.
Hey, thank you, Christina. My name is Kasahoon, and it is my pleasure to introduce Evan Wood today. He is a PhD candidate doing a doctoral program at UCSD and SDSU in the chemistry. He is working in Dr. Manal Suerjo's lab, where they focus on biochemistry, X-ray crystallography, and cryo-EM. And he is working with DpD system, a recently discovered restriction modification system, which he will talk to us about in a few minutes. A few of the positions and honors he held, in addition to the graduate fellowship, he is a recipient of California Metabolic Institute Irene Wick Fellowship. He has presented at multiple Gordon conferences. He is a recipient of ParB's Biomedical Research Endowed Scholarship, Michael and Donna Maley Endowed Scholarship, Harry E. Hammer Memorial Scholarship, and so on. He's been to NCCAT before through the GUP3 program, where he optimized cryo-EM sample preparation conditions for his samples. And his future is to pursue tenure position at an R1 institution where he will be continuing research in nucleic acid. So Evan, thank you for accepting our invitation, and we look forward to your talk.
Thank you for that introduction, Kasan. The real thank you should go to you and Eugene for helping me when I went there to screen these samples. Okay, does this look good? Let's see here. Oops, 1 second here. Okay, thank you everyone for coming to the webinar this morning. I'm excited to show you sort of the results that kind of was the apex of my PhD. So what I'm going to go over today is, first I'm going to go over the system I'm studying, which is the 7-diazepyridine and DNA system. Then I'm going to go over how I optimized the sample and grid preparation to overcome some issues that I was having. And then finally, I'm just going to show off the nice density and the structural results and what I was able to learn about my system from cryo-EM. Okay. So first off, my system is a restriction modification system. And just for a little background, a restriction modification system is like the bacteria's immune system against phage, right? So you have these restriction enzymes that recognize these palindromic sequences in DNA, So when the phage infects the bacterium, it'll inject its DNA, and these restriction enzymes will chop up the phage's genome. The problem with that, though, is that those sequences inevitably also exist in the bacteria's genome. And so it needs to shield its own genome from these restriction enzymes. And so it has these modification enzymes, which will modify these sequences and protect it from restriction. And on the left here, I just have an electron micrograph of some phage infecting bacteria, which I thought was pretty interesting. It was taken at UCSF. So a little more background on restriction modification systems. On the left-hand side of the screen, I have the traditional, the older, more well-characterized types of restriction modification systems. These were pretty much all methylation-based. And one thing I want to highlight is genetically, they were made up of a few genes, one, two, maybe three, restriction gene, modification gene, specificity gene. But my system, and more recently discovered 7-deoxyguanine based system, is much more complex. The modification system consists of 3 genes alone. And then the restriction portion of the system consists of, we think, 8 genes. And then there's also this other very interesting modification system, the phosphorothioate system, which I won't be talking about today, but it was also discovered around the same time, I think, and also pretty unique and complex. And there is some homology between a couple of the modification genes between those, but not too much. Another thing I want to highlight is sort of the chemical diversity of 7-deoxyguanines. So 7-deoxyguanine modification system. Originally, they were sort of found in tRNA. And people were kind of guessing or hypothesizing for a long time that they could also exist in DNA, partly because of this existence of there's this TGT-like or this DpdA gene that was being found in phage, and this TGT system modifies tRNA. So it didn't quite make sense for the phage to have those. And so they thought maybe it's modifying the DNA. And I just lifted this figure on the right from collaborators' paper because I really liked that it's a pathway figure showing deoxyguanine modifications in DNA and tRNA, but I just really like that it highlights all the different kinds of 7-deoxyguanines that can be found nowadays in DNA and tRNA and various species. So like I mentioned earlier, DpdA is homologous to these tRNA-guanine transglycosylase, or TGT for short, system. On the top left, I have the crystal structure of DpdA. And in the top right, I have the bacteria crystal structure of TGT. And then bottom right, the archaea structure of TGT. And then bottom left, the human structure of TGT. In gray is the sort of conserved tin barrel folds that all of these proteins share. And in green are the unique insertions relative to the tin barrel fold that each protein has. And for DpdA, there's these 3 major insertions that set it apart from the TGTs, just from a sort of a broad view. You have insertion 8, which is a gigantic alpha helical bundle. You have insertion 1, it's a long loop, and then you have this insertion 2. And we didn't know what those were doing for a long time, but now, thankfully, with the same structure, we do know. And then on the right, where these enzymes really show to be dramatically similar is the active site. So they all will have these 3 aspartic acids. In my system, it's 256, 95, and 130. And then they also share these specificity loops. Now, these change depending on which 7-deoxyguanine in the transglycosylase site is using. And it's pretty remarkable that, the human, I really like the human 7-deoxyguanine with this giant functional loop on it. But anyway, so what do we already know? So we know that DpdA and DpdB work together to install this PreQ0, this precursor base in DNA. It's this 7-deoxyguanine with a cyanide group on the end there. We know that the sequence that it recognizes specifically is the CAG-CTG sequence. And we know that DpdA is the transglycosylase because of its homology to GT. And basically it excises this central guanine and replaces it with precursor. But what we don't know is how does the system discriminate between all of the different DNA sequences in nature? How does it find and only modify the CAG-CTG sequence? And also, what is DpdB doing? It's an ATPase. We know it hydrolyzes ATP, but that requirement is not found in the TGT system. The TGT system only requires the transglycosylase. So it was pretty interesting to find that this system also needed this ATPase. That's sort of this mysterious ATP activity. So that's kind of the two goals of what I'm trying to investigate here. I decided to pursue cryo-EM, and it was immediately apparent to me that, in my system, there's several macromolecules involved, and there's potentially dozens of different ligand options I can choose from, or lengths of DNA. And then when you start including the formulation of the construct as well, such as, additives and maybe cross-linking agents or how you will prepare them, the number of different variables, it just becomes endless very quickly. And so I set out to find a way to rapidly screen in-house different sample conditions so that I could just, when I do finally get that cryo-EM time, I can just come in with the best, most well-characterized, the best and most stable sample available to me. So this ClarioStar is equipped, in our lab, is equipped with the lasers and filters necessary to perform fluorescence polarization experiments. Now, briefly, fluorescence polarization is, the way it works is you can have this sort of DNA duplex. It's tagged with fluorophore, and before being bound by the DNA, by the protein, it's smaller, it's tumbling faster in solution. And once these enzymes bind the DNA, it will tumble slower in solution, and that produces this polarization of the fluorescent signal, and that's a measurable effect. And so on the left-hand side of the screen, I have the sort of the KDs, the dissociation constants of DpdA and B for DNA under various conditions. And this was sort of a major result for me because originally I was actually using these native gels, which were not really showing the same result, but this is a continuous assay. And so it allowed me to measure more accurately what was going on in my system. But what I noticed is that ATP hydrolysis really seemed to be vital. It seemed to be really important for high affinity binding. For DpdB alone, it seemed to have, at least a 4-fold or a 3-fold increase in affinity. But if you had excess DpdB and then you just titrated in DpdA's affinity for the DNA, of course, with ATP present, was now, multiple orders of magnitude higher. And so that told me something important is going on there, and likely I need to have ATP in my sample for whatever structural studies I'll be doing. And then on the right, I have a sort of dissociation kinetic assay. And this kind of just highlights the requirement for ATP. In blue, I have DpdA and DpdB. And so briefly, in this assay, the way it works is I have some pre-bound fluorescent DNA. And then I use the ClarioStar, which has these syringes and tubing that can inject into your 96-well plate. It will inject, large excess of unlabeled DNA. And so what we're monitoring is just the dissociation of the enzymes from the fluorescent DNA. And so in blue, I have DpdA and B without ATP. And in fact, the half-life is so short that you can't even measure these proteins dissociating from the DNA without ATP. The dead time of the instrument is 1.8 seconds. And so I didn't measure anything. So the half-life is potentially much lower or just a little bit lower than 1.8 seconds. But once you add ATP, still relatively short half-life compared to maybe other systems, but now it's measurable, 30 seconds half-life. Once you add pre-K0, the natural substrate for DpdA, it's shorter, which is what you'd expect because now you're measuring these enzymes dissociating from what would be the product. And then you have DpdAB ATP and this 9-deazoguanine molecule. And I'll go into a little bit more detail about what that's doing in later on in the presentation. But essentially, that is covalently trapping DpdA on the DNA. So in fact, it never really sort of plateaus down at 0 down here. And the half-life is considerably longer than the length of the experiment. Another clue that ATP and DpdB were really required for sort of high affinity binding for me, or for forming this interesting construct that is actually recognizing the sequence, is that I actually crystallized DpdA bound to DNA without DpdB or ATP present. And it was just interacting with the phosphate backbone. And in fact, it had this weird structure where sort of the ends of the DNA are stuck into the active site. And that's partly because there's a sticky end on the ends of these DNA. And that flipped out base is sort of wiggling itself into the active site. But there is no recognition, no readout, no interaction with the central sequence. And so all these results combined really told me, okay, I need to add ATP, I need to have DpdB, I need to have everything present. And that probably makes this not really suitable for crystallography, and I should pursue Cryo-EM. So in the top left here, I have a micrograph from one of my very first Cryo-EM experiments done at UCSD using a AU flat that we had. It was all we had available to us at the time in our lab. And one thing I want to point out is just how noodly the micrograph is, how spaghetti-like it is. You can see all this, strands of DNA, and that depending on your system, that may be good, that may be bad. It turns out for my system, that's not exactly what I wanted. I didn't know that at the time, and I collected a small data set, and I processed the data, and it turned out that basically 99% of my complex was actually dissociated, and so I mostly had just a bunch of free-floating DNA and proteins, but nothing was interacting, and that was really disappointing for me. But I did see this one class, that really gave me hope, and I now know what that is, and that's DpdA bound to DNA. And then I went to NCCAT in New York, and they allowed me to screen various different grid types and other conditions. We weren't really sure what was causing the dissociation. I screened all sorts of things, different sample conditions. I screened detergents. I screened some cross-linkers in-house. I tried doing SEC first, right before making the grid, and nothing really seemed to be alleviating this dissociation. But when I went to NCCAT, they had all these grids available to them, and they allowed me to use this QuantiFlow grid. And immediately I noticed that there's a difference between the micrographs. This looks more globular. It looks, somewhat heterogeneous, of course, but it definitely, I'm seeing a major difference between these two micrographs. And I actually saw some difference in sort of complexes on the DNA. We had a small data set here. And one thing I want to highlight, what I now know is that this sort of gray protein on this 2D class, this, shifted, this other protein in this 2D class here is actually DpdB. And so for some reason that was not showing up on the AU flats, but now on the QuantiFlows it was. And what actually ended up being sort of the grid type that was the best for me were these graphene oxide coded QuantiFlows. Now, maybe a regular QuantiFlow full data set might have worked, but I noticed that there was less of this dissociation on these graphene oxide grids. And the contrast and overall appearance of the atlas of these grids was really nice. And so I decided to collect a full data set on these. And what I have on the top right here is these 2D classes from the final sort of particle stack for the map I'm going to show you later on. And so you have these really clean 2D classes. And then in the bottom, I have 2D classes highlighted in various colors and their corresponding particle picks that I found using the inspect particles job in CryoSpark. And then, and then one thing I wanted to mention is that these graphene oxide grids were commercially sourced from mitogen. So clearly the sample was heterogeneous, and once I collected a full data set, it definitely reflected that. But instead of having a bunch of dissociated protein and DNA, what I actually seemed to have was a variety of different DNA complexes, which is for me, a better problem to have, but still it required some extensive processing to sort out. And so on the left here, I have some 2D classes of various different species that I observed on the grid. So this central DpdAB and DNA class is representative of sort of the final map and I think the biologically relevant complex. And then here on the right, some 3D volumes that I sort of just made from these other species that I found that helped me sort out the particle stack by heterogeneous refinement. So other than that sort of compositional heterogeneity, another thing I noticed was this continuous heterogeneity once I got to sort of the final stages of processing. And then it's largely from DpdB. And so really quickly, what I have here in the darker blue is DpdA. And then in the lighter bluish green is the DpdB. But what I actually was only able to model was the C-terminal DNA binding domain of DpdB. But there was this sort of blobular, poorly resolved density that was in approximately the correct position for the N-terminal domain of DpdB. And I think it's probably just flexing around partly because at least, AlphaFold predicts there's sort of this long sort of linker between the two domains. And I have some other data from some SAX experiments suggesting that there's some conformational changing going on. And so unfortunately, I was not able to resolve that, but it was something to consider. So to overcome this heterogeneity, I went with a pretty classic data processing strategy. I started off with blob picker, just sort of because I didn't have any sort of good idea of what my sample would actually look like on DNA. And I just sort of did these iterative rounds of 2D classification, multi-class ab initio, followed by multi-class heterogeneous refinement to sort these particles out. Importantly, though, in sort of the earlier stages, in the earlier iterations, I was more conservative during the 2D classification of which classes I kept and which I tossed out. I would toss out only the most obviously poor classes, like ice or some weird-looking particles. And then I would loop some particles back together that were misclassified in 3D. And then in the last iteration, I was very aggressive and only selected the highest quality 2D classes. And I used that to generate a volume, and that volume to generate templates. And then those templates allowed me to do template picker, which actually was sort of one of the major improvements, because now I started off with a cleaner particle stack, but it also seemed to pick up different orientations and different views of my particle that I may have been missing before with blob picker. And then again, I did this iterative 2D and then 3D sorting, but with a key difference being that after 2D classification, I just immediately went into heterogeneous refinement from what I had obtained earlier. I did decoy heterogeneous refinement with all of the species volumes that I observed that I could, obtain, and as well as a couple junk volumes. And that allowed me to sort of retain a lot of these high quality and quickly sort a lot of these particles, and that generated this volume that you're now seeing. So on the top, you have, again, DpdB, and the bottom, you have DpdA. And you have a pretty nice map. Towards the ends, obviously, it's getting a little bit lower resolution, but what we care about is all in here. And so again, here's the map colored by local resolution. So in the middle regions is the is where the active site is, and is thankfully where the highest resolution also happens to be. And it also happens to be where that sort of central CAG-CTG sequence is. And so that allows me to resolve everything I want to know about how the sequence is being recognized and what's going on in the active site. And then now I'm just going to sort of go through the actual, what we actually learned biologically. So on the top left, I have the electrostatic map of A and B and the DNA, and you can see how they fit together to sort of make this nice positively charged groove that perfectly accommodates this DNA. And then you see in the middle, this central base pair is broken, and that sort of substrate guanine is stuck into the active site, and opposing it is cytosine is stuck into its own pocket. Here I have just highlighted the just the degree to which DNA is being bent. And then on the right-hand side of the screen, what I have shown, these sphere side chains are the residues involved in direct base readout. So DpdA does seem to have this direct base readout mechanism, meaning it's just hydrogen bonding with the Watson-Crick, on the Watson-Crick interfaces in the major groove of the DNA. And I really like this bottom figure because you can see from the bottom of the DpdA, you can see how just these proteins sort of trace the major groove from left to right along the active site. And so here's sort of just an overall map of the complex. And I've color-coded the DNA here by base pair. One thing that was obvious to me immediately is that DpdA was doing all of the base readout. DpdB was just sort of in the back here. It was bound to DpdA, and it was also interacting with the phosphate backbone of the DNA. And so maybe helping bend and stabilize that DNA, but not really reading any sequence, at least in this stage of the reaction. And so the sequence that we're of interest here is this essential immediate, the 6-base palindrome in the middle, the CAG-CTG sequence. But I also noticed a couple bases on the edge that seem to be being recognized by some side chains of the proteins, and also some activity data from some collaborators, which I don't have shown, is suggested that maybe these also have some kind of an effect on the activity of this system. And so now, since we are all structural biologists, I know you want to see the density. And so here I just have shown, sort of starting from the right, the purple and pink base pair being recognized by this K66. And then I also have in the top right an activity assay that I did where I mutated to the lysine to an alanine, and indeed this lysine is important. And then moving closer to the active site here, now we're looking at the sort of central light and blue base pairs. And these seem to be recognized by arginine 390 and aspartic acid 13. And one thing I want to highlight here is, the density is of high enough quality to actually be able to discriminate between purines and pyrimidines. And also I was able to distinguish sort of the polarity of the DNA, because that was not immediately clear to me how that would be fitting into the DNA, or how the DNA would be fitting onto the protein for this result. But the density did allow me to discriminate. And so now what we have shown is actually the actual active site. Now in this active site, this is actually 9-deazoguanine. So that guanine, that substrate guanine, has already been excised. And now in its place is this 9-deazoguanine. And again, we have those 3 important aspartic acids shown here, 95, 256, and 130. And we have some other, here's a specificity loop. It's not doing anything because this is not a 7-deazoguanine. And then you can see sort of the phosphate sugar backbone sort of arching into and then back out of this active site. In the top right here, I mutated these sort of key aspartic acids and show that these are pretty much catalytically dead. And so I wanted to go over exactly why this 90-as-guanine is helping my system. Now, this is something that we actually learned from the TGT people because they used the same sort of trick to trap TGT on tRNA and stem loop RNA for crystallography and for cryo-EM. And so what you can immediately notice here is that there is this continuous density between this ribose and this aspartic acid 256. And the density between between this guanine and the ribose is broken. And that's because this is actually forming a covalent bond between the C1 carbon and the aspartic acid. And the bottom here, we just have the reaction mechanism, which shows why this works. So you have aspartic acid 256 acting as the initial nucleophile that will, help kick off that substrate guanine. And then preQ0 is supposed to come into the active site, is activated by aspartic acid 95. And then the ninth nitrogen on preQ0 acts as sort of the final nucleophile that reattaches this nucleobase, or that attaches this nucleobase to the ribose. But what we have going on here is what you can do is if you just have large excess of 90-as-guanine present, the initial portion of this reaction where will still occur, where that guanine will be excised, but this 90-as-guanine can outcompete the original substrate guanine for the active site and trap the protein covalently bound on this DNA. And so what we're really observing is sort of this stage of the reaction. That's what's going on in this structure. Okay, and then back to sort of base readout here. I have some density and showing the pocket for the cytosine that was originally base paired to the substrate guanine. I have a couple mutants in the top right as well. And then we have this yellow base pair, again, more base readout. I liked this. This was a very interesting sort of structural result for me. So this, by the way, this Q262 is actually hydrogen bonding with something off the screen here. So I know that the orientation looks a little off. Right? That's because it's interacting with something else down here, but I still think that it's forming this sort of this U-shaped structure that kind of accommodates this base pair here. And then lastly, we have sort of the red and orange base pairs being recognized by arginine 268. And one thing I wanted to mention is that once pretty much immediately outside of this sort of central palindrome, the density of the base pairs quickly deteriorates. They become more smeared together in both directions. And I think that is a couple different reasons. I think that could be because the DNA is more flexible the farther you get from the active site. But also, I think it's possible, since this is a central this palindromic sequence, technically the DNA could be oriented, in two different ways. This guanine may be being modified, or this guanine, or both. We're not really sure what's going on yet, but there is, based on the structure, there's not really any way that I can tell for the system to discriminate from one strand versus the other strand. And so outside of that central base palindrome, where it's no longer palindromic, you have base density that's being averaged with other bases that are not the same. And so another thing that I wanted to quickly go over that we are sort of learning from the structure is what is DpdB doing? Because that was the other major goal besides understanding sequence recognition. It does now seem that, yes, DpdB and ATP somehow sort of loads DpdA onto that central palindrome. Unfortunately, how it does that is still kind of a mystery to me. But another sort of curious thing that I observed is that I only observed really a monomer of DpdB, or half of a monomer of DpdB in this data set. But I've characterized DpdB in solution through multiple biophysical methods. So on the right here, I'm showing some small angle X-ray scattering data. And I have the theoretical molecular weight from a monomer, as well as the calculated molecular weight of DpdB in solution from SACS. And then I also have sort of the AlphaFold model for a dimer of DpdB, along with an envelope that I calculated from that SACS data. And using this sort of conformational sampling software that samples a variety of conformations and fits that to SACS data, I was able to show that I think that there's probably an open and a closed form of DpdB, if assuming this is the correct structure from AlphaFold. I mean, it fits the SACS density pretty well, but that, is a pretty dramatic departure from what I'm observing by cryo-EM. I also characterize this by analytical ultracentrifugation. And so, again, in solution, DpdB seemed to be a dimer. And with DNA, it also seemed to be a dimer. Now, one caveat is that I didn't have ATP in this AUC experiment, and I may go revisit that and add ATP and see if that changes anything. But I did do some SACS experiments with ATP, with DpdB in solution, and those still showed that it was a dimer. So it's possible that perhaps in the cryo-EM data, DpdB is still a dimer, and I'm really only observing I'm missing 3/4 of it, because the dimerization domain is in this N-terminal ATPase domain, this top portion of DpdB. And maybe that is sort of flexing relative to this DNA bound domain, and it's completely non-observed, or maybe the stoichiometry in the reaction changes. I'm not really sure. And then lastly, a little bit more about DpdB. So after it initially seems to load DpdA onto the DNA, it seems to have this sort of maybe secondary role where it's interacting with the DNA by phosphate backbone interactions with the minor groove. So I think it's helping this sort of dense DNA conformation. It's helping stabilize that, maybe during the length of the reaction. And so, since, one thing that makes this system really interesting is when you're comparing it with TGT, because, the sort of central base exchange, its transglycosylase mechanism is basically the exact same in both systems, and they both work on nucleic acids. And so it's interesting as a biochemist to sort of compare and contrast between the two of them. And so on the right, I have the cryo-EM structure of the TGT, of the human TGT, that was published, I think, a couple years ago now. And on the left, I have my structure. And so these were actually superimposed in Primal, but then I shifted them apart so that you could more easily distinguish the differences between the two. So one thing that was, immediately obvious to me is once you leave the active site, there seems to be almost no changes when it comes to, there seems to be almost no similarities when it comes to the nucleic acid binding mode between the two. In fact, they're almost perfectly perpendicular. Also, DpdA, unlike the TGTs, did not really seem to be a dimer. I did mention earlier that there were other DpdA molecules. Some of the sort of contaminant species that I observed was 2 DpdAs on the DNA, but that second DpdA really seemed to kind of just be not really interacting with these other parts of the complex and kind of just off on its own on the edge of the DNA. So I'm still not. I don't think it's involved in the reaction, but I'm still not entirely sure. And again, of course, the active site between the two enzymes, or the two transglycosylases, it's more or less completely conserved. So that's pretty much it for me. In summary, I optimized sample conditions cryo-EM, largely through in-house biochemical screening first. And I think that was really necessary because there's just way too many different conditions that you could test by cryo-EM. And if you had to test every single one of them by cryo-EM, that would just be prohibitively expensive and time-consuming. Additionally, I found that it seemed like the sort of major improvement in my system was identifying the proper grid type for specimen preparation. It seemed like these AU flats were almost toxic to my sample. Now, that's not a general, I'm not going to say that's a general rule for nucleic acid systems. It's not. But I will say that my colleague, Isaac, who's also in my lab, is working on another part of the same system, but it is a completely different fold and a completely different enzyme. It's DPC, and it also works on DNA. And he noticed the same thing with the AU flats, that um, his sample was also just completely dissociated. But also my other colleague, Anna, she was working on a tRNA system, and her complex was just fine on the AU flats. She did require some sample optimization in the form of tRNA folding to improve that sample so that she could get it associated, but the AU flats had no effect on that front. And then, the graphene oxide coated grids really made sort of the best grid for me. And then the actual sort of biological results are that DpdA is recognizing that CAG-CTG sequence through a direct base readout mechanism. And now we have a structural basis for how that works. And also that B and ATP hydrolysis really seems to be driving this sort of sequence recognition somehow. I'm really curious as to how that is, and I hope that in the future I'll get another chance to sort of go back and maybe do some more cryo-EM experiments with DpdB by itself, or with other ATP analogs, to sort of discriminate what exactly it's doing there. And that's it. I just want to say thank you to everybody involved, my PI, Manal, 3 sort of students in my lab who were key. They gave me advice, they gave me some support. George did some of these fluorescent polarization experiments that I showed you earlier for me. He's an undergrad in our lab. I want to thank my collaborators for meeting and discussing with me multiple times, and guiding me through this process. And most importantly, I want to also thank all the people who took their time to teach me cryo-EM, all these specialists. Eugene, who I don't know if he's here today, and Kasahan at NCCat were sort of vital. I really appreciate their approach when I went there to screen my samples. They recognized that I had a little bit of preliminary data and that everything was just completely dissociated and that we needed to make some very large improvements. So we screened a whole bunch of different conditions, all kinds of crazy stuff, not blood discharging grids, detergents, graphene oxide grids, different constructs. And so that really allowed me to move quickly, and because we needed to explore a sort of a wide space of different conditions. And then I also wanted to thank Nathan Burrows at S2C2. He's not here today, I don't think, but I did send some of these AU flats their way, and he did collect some data for me. And while those experiments weren't the ones that ended up working, they did help me narrow down and get to sort of the final condition that worked out for me. And of course, Mar at UCSD, he's the facility director at the Cryo-EM facility at UCSD, and I went there and he taught me how to use the scope, and he also gave me a lot of advice and data processing and data collection. And then lastly, Dr. Shane Gonan at UC Irvine. I kind of just want to give a shout out to him because he actually told me, he called it like a couple years ago. He gave a talk at SDSU, and I told him I was having this sample dissociation problem. And he mentioned, he's like, hey, just try different types of grid. And I wish I listened to him sooner, but I was reading all these review papers and it seemed like grid type was sort of lowered down on the list of things that people try initially to help resolve this dissociation issue. And that's it. And I'd be happy to take any questions.
Thank you, Evan. That was fantastic. Sorry, you always wish you took the early advice that turned out to be right, because hindsight is 2020. But
Of course. I mean, I think everyone's system is different, and it could have been any other thing. It's hard to know what to do first.
Exactly. And I'm going to start with my own question because I can. So you see this covalent linkage, right? Which then makes you wonder why this thing is falling apart on the grid. And it's very, very normal to see these things falling apart on the grid. That's not unique.
Right.
So that makes me wonder how many, like what percentage of the things you're picking off the grid ends up in this class where you have this covalent interaction?
That is a good question. If I had, higher resolution data for all of the other species, I would be able to tell you more about that. I can tell you from experience that um, one key thing about this is that, it's not like I'm like sort of cross-linking this. This is not like some sort of irreversible covalent bond. I am sort of just out competing that guanine, that original guanine that has that nucleophile that will destroy this bond with a reverse reaction. And one thing that I noticed compared to the TGT people, which seem to have a much easier time forming this, very early on in my PhD, I was reading their papers and they actually confirmed the existence of this covalent bond through SDS page, which is a pretty harsh condition. It's a denaturing condition. You've got a whole bunch of detergent. You've got, a ton of reducing agent. And well, actually, I think they maybe sort of didn't include the reducing agent, but they showed that there was a tRNA protein bound band even under these denaturing conditions when you added 9-deoxyguanine. And so I tried reproducing that with my system, but I never was able to do it by SDS page. But it was this sort of continuous fluorescence polarization half-life determination assay that I kind of, explored that really gave me the confidence to pursue this. So I think, it is helping stabilize the reaction, but it's not like you can still dissociate from this. In fact, I know that I tried like these experiments where I added like 200 millimolar salt, and for some reason that still dissociates this complex, even though it's covalent. It's like, I mean, it is covalently bound, but it's, so why that is, I'm not entirely sure. You wouldn't really expect that, but yeah.
Thank you. Actually, so you mentioned the fluorescence polarization, and there was a question actually about that experiment. What was the size of the DNA you used for the fluorescence polarization?
Good question. It was a 40 base pair DNA. And so that wasn't chosen. I mean, it was originally chosen somewhat arbitrarily, but there is some data to kind of back that up that I didn't show here, which is we did some collaborators of ours did activity assays on various lengths of DNA, and we found that the 40 base pair had really good activity. But once you went below the 40 base pair, you started to see kind of a drop off in activity. It's not entirely clear exactly how short you can get yet because we didn't thoroughly go through that. Maybe 38, maybe 36 base pairs would be enough, but 30 and 20 base pairs seem to be, you seem to get a noticeable drop in activity. Now, so that's kind of another interesting thing about this system is because we thought that because 40 base pairs is had the highest activity, that whatever proteins could fit, whatever the stoichiometry could be to be on that DNA would be sort of the biologically relevant stoichiometry, but that actually didn't really seem to be the case. So I think I would be wary of, overly trusting these biophysical results when you're working with sort of DNA binding systems. Because I actually had a lot of results that showed DpdA would dimerize on the DNA, but there's just another DNA strand that DpdA was kind of just non-specifically interacting with corner.
All right, we have a number of questions about sort of your data processing. First, there's a question about what software you use for refining nucleic acids.
So everything here was done in CryoSpark. And I didn't really do any sort of super fancy softwares or anything like that, where you have to install them and download them from GitHub or anything like that. I did. One thing I will note is, so for the final volume, I did non-uniform refinement, which is, I think, pretty standard nowadays. One thing I tried initially in the beginning with picking with blob picker is I did both elliptical and circular blob picker, and then I combined the results and I discarded the duplicate particles. And that seemed to pick up more particles. Now, in hindsight, I'm not really sure how much elliptical helped because the DNA is not really elongated in my system. But my colleague Isaac, the DNA is not very extremely dense, and it's still more or less linear. And I think it might help pick up some of those particles.
Right, actually, a couple more questions about your processing. For successive non-uniform refinements, is it allowed or recommended to perform multiple successive non-uniform refinements in CryoSpark on the same data set, or is non-uniform refinement something you should only do once at a certain stage?
I mean, so I'm not some data processing guru here. So I can tell you what I think, but take that with a grain of salt. I personally didn't really like if you just take the same particle stack and you just do multiple rounds of non-uniform refinement, it didn't really seem to change too much for me. Now, if you start changing up what that particle stack consists of, now that is going to have some kind of result. And if you start doing interesting things with masking, you can also sort of change up the results of that non-uniform refinement. Yeah. Oh, one other thing that I would mention, just kind of like an observation I made is when you are doing 2D classification earlier on, it is sort of difficult, especially if you have a heterogeneous system like mine, to kind of tell what you're looking at. And so I did notice a lot of these like very small, sort of like bright white circles or spheres. And I was like, what is that? And it ended up being sort of the DNA sort of pointing into the camera. So if you're looking at that and you're thinking it's ice or some weird thing, I wouldn't necessarily discard it. I mean, if it's some giant white circle, that's ice. But if it's some small little bright white thing, it's probably the DNA sort of pointing straight into the camera.
All right. And then another question related to non-uniform refinement. So can you use an ab initio volume from an earlier stage of processing to do non-uniform refinement on a then more finely curated final set of particles? Or is it better if you have this more finely curated particles to generate a new ab initio before then going on to additional refinement?
Wait, I'm sorry. Can you repeat that one more time for me?
Yeah. So if you have a volume from an earlier stage of processing,
Mhm.
and now you have a more finely curated final set of particles, and you want to do non-uniform refinement using that curated set, should you generate a new ab initio from that set, or is it okay to use an earlier volume from earlier stages of refinement?
Oh, I think I understand. Okay, so first off, I don't think you can input an ab initio volume into a non-uniform job because I think you need what is it, the half maps from sort of a refinement job. Not entirely sure about that, but if we're just talking about does it matter what volumes you input? I mean, I think you need to input something that's kind of somewhat close to the sort of final volume you're looking for. But what really makes the difference is, I think, the particle stack. But yeah, I think you do kind of want to have an earlier some sort of heterogeneous or homogeneous refinement job to kind of sort of hone in on sort of the final structure and help with because if you start off with like an ab initio, well, yeah, you need a mask, so you need some sort of heterogeneous refine or homogeneous refinement job before you can do non-uniform refinement.
Thank you. We have a couple more questions. If you don't mind sticking around, we'll get to them.
Yeah,
But just
totally.
for those of you who have to head off at the end of the hour, thank you for joining us today. We wish you all well in your cryo-EM journeys, so let us know how we can help you. If we didn't get to your question and you have to head off, come back to find the recording. You can find it through cryomcenters.org. So thanks for joining us, and we'll stick around for another couple of minutes to wrap up these last questions. All right, so there's a question about using the graphene oxide grids. Did you need to lower your sample concentration when you move to graphene oxide?
That's an excellent question. And, in hindsight, I think it does look, I don't know if it's necessarily overcrowded. It's okay. I think if it's crowded a little bit, as long as the particles aren't overlapping. I maybe could have decreased the sample concentration a little bit compared to what I did, but in reality, it was actually one of the things that Eugene and Kasan had me try initially. Is I used a considerably lower concentration compared to the other grades, and we didn't really see anything, and you know, and so we were kind of surprised, and but I pushed them. I'm like, all right, come on, let me just try one with the same concentration and see what happens. And that actually seemed to be the case. Is the same concentration, more or less, from these quantifiers was working for the graphene oxide grids. One thing I would be speaking of graphene oxide grids is one thing I'd be cautious of is they're not always uniformly sort of created. A lot of the squares maybe absent of graphene oxide, especially we bought some graphene oxide grids after this experiment. But then I went to UCSD and I screened them, and Mar put it in diffraction mode, and we were looking all over the grids, and it only seemed like around 2% of the surface was covered in graphene oxide. So we kind of got, so you want to be cautious about, are you actually looking at a graphene oxide coated grid or are you not?
There's actually not a question, but a comment from Dennis, who says grid type also made a huge difference with complex dissociation problems for them. Graphene grids worked when gold quantifoil graphene oxide did not. So
Interesting.
Another vote for team try different.
Was it a nucleic acid system? I'm just out of curiosity.
Dennis, if you're still here, if you want to follow up,
I hope so.
Let us know.
Yeah, I've long wondered if this gold is somehow bad for if it's nucleic acids, or maybe in my system it's just DpdB. I'm not sure.
And actually, there's a comment. No, just protein, non-nucleic acid. So
okay, so who knows what's going on.
And then one last question. Have you tried cross-linking? I think you mentioned you did do some cross-link experiments, but they mentioned specifically BS3 has worked for them.
I did do BS3 actually. So one caveat is I never actually put any of these cross-linked samples on a grid because we had limited EM time, and I screened them in-house. I cross-linked them, would run them on an SES page and a native page to confirm that A, it was cross-linked, and B, the DNA was also still bound. But it seemed like in my case, with most of the cross-linkers that I tried, the DNA would be dissociated if the protein was cross-linked, or the protein wouldn't be cross-linked. And I think I attributed that largely because a lot of these cross-linkers react with the primary amines, so these lysines. And in my system, these lysines are important there's a lot of phosphate backbone interactions from these lysines. And so if you get rid of those, you just completely destroy binding.
It's nice when things don't work, but then it makes sense why they wouldn't.
It never makes sense until the end.
Right.
But it is nice, yeah.
All right, we have gotten to all of the questions.
Excellent.
So thank you so much for sticking around and getting to those. I'm going to go ahead and stop recording.